Hey guys, this is me, Dr. L, and we're going to talk about lipids, especially focusing on phospholipids so that we can better understand the chemistry and therefore the function of cell membranes. Here's a drawing of a phospholipid. So over here on the left is the phospholipid, and the top part that's in a ball is referred to as the phosphate head, and it's called that because it contains a phosphate functional group, which is shown in lighter yellow here in a slight yellow circle. That is a very polar group. In other words, it will have a tendency to hydrogen bond with water. How do we know that? We see a full negative charge on this oxygen, and also if you look up the electronegativities of the phosphorus atom and an oxygen atom, their electronegativities are quite different. Oxygen's extremely electronegative. It's going to pull electrons that are shared between these two, the phosphorus and the oxygen atoms. Those electrons are going to be pulled towards the oxygen. So where we see these covalent bonds in blue, those are very polar covalent bonds. So there's a high tendency of this phosphate group to form hydrogen bonds with water. That makes it water loving, which is called hydrophilic. Okay, and so that's really, really important to remember. Because of that phosphate group, this entire phosphate head is going to tend to hydrogen bond with water. It's going to always orient towards and interact with water molecules. Where are there water molecules on the cell? On the outside of the cell and on the inside of the cell. That phosphate group is attached to what was a glycerol molecule, and that is bonded through chemical bonds called, these are ester bonds, to two tails. And the tails are called fatty acid tails, and they are extremely hydrophobic. So up here on the right, we have, it's a little bit faint, but you can see these long hydrocarbon molecules. And these are examples of what fatty acids can look like. So fatty acids are a type of lipid. They are always going to consist of a chain of carbons covalently bonded together. Carbon-carbon bonds are nonpolar covalent bonds because the electronegativity of a carbon matches the electronegativity of a carbon. And in all fatty acids on one end, there will be a group called a carboxylic acid group. That group goes away in the formation of the ester bonds that join these fatty acids onto this glycerol backbone to form an intact phospholipid. So we're looking at these little building blocks here when we look at fatty acids. They're part of the larger phospholipid molecule, but they can also exist just as molecules on their own in cells. Fatty acids have a lot of variability in their chemistry. So things that can vary is the length of the carbon-carbon backbone. Another thing that can vary is the number of hydrogens that are attached to the carbon-carbon backbone. So on the top, we see what is called a saturated fatty acid, meaning that every carbon there is single bonded to something, either a neighboring carbon or a hydrogen. So we have the maximum amounts of hydrogen possible in that stearic acid shown on the top. In the bottom fatty acid, it's an example of an unsaturated fatty acid. Unsaturated fatty acids contain some carbon-carbon double covalent bonds, and where carbon-carbons have double covalent bonds, they're going to be bonded to fewer hydrogens because each carbon has a valence of 4. Therefore, it can form 4 covalent bonds with something else. And so if you've got a carbon-carbon double bond, it can only have 2 more single bonds. So that reduces the total amount of hydrogens, which changes the chemistry of the fatty acid. And what we see is those double bonds will introduce a bit of a bend to the molecule, which is what we see in the fatty acid tail that's shown on the right in the intact phospholipid molecule. And those little kinks can actually change like how phospholipids move and orient in a membrane. So that matters, and we'll get back to that later. So some of the questions that I've written for you here on this slide are: what kind of chemical bonds are holding together the fatty acid tails, and why are these parts of the phospholipid molecule hydrophobic? The chemical bonds that I'm referring to are the carbon-carbon bonds and the carbon-hydrogen bonds. Those are very nonpolar covalent bonds. Therefore, all of these molecules along their lengths tend to be very nonpolar. They will not hydrogen bond with water, which makes these fatty acid tails in the phospholipid molecule hydrophobic. Okay? The second question is, why is the phosphate head hydrophilic? And I answered that a moment ago. The third question is, what is the particular name for the chemical bond marked with an arrow? And up here we've got a chemical bond marked with an arrow, and it's a little blurry here, but that's a C double bond to an O. That is a very sorry, I'm actually not referring to that one, not the red one. What I meant to draw your attention to is the black arrow pointing over on the phosphate group, and I'm realizing now that black arrow kind of shifted down a little, so I just moved it up. And that is an ester bond, which is a characteristic type of bond that frequently shows up in lipid molecules. Phospholipids are a category of lipid, as are fatty acids. All right, so a little more on lipids. Lipids are one of the 4 categories of macromolecules that are important in cells, the other 3 being nucleic acids, carbohydrates, and proteins, right? So when we talk about membranes, we tend to focus on lipids, and by focusing on phospholipids. There are other types of lipids, though. So that phospholipid is drawn in the middle of this figure. On the left is a drawing of something called a triglyceride, and you can see how it's constructed kind of similarly. The blue or purple ball here would be the phosphate head, and then these 3 green, like tail-like structures would be fatty acids. And over on the right, also considered lipids are steroids and waxes. An example of a steroid is cholesterol. And an example of a wax is paraffin wax. I'll show you a picture of that in a moment. But both those categories of molecules are lipids. Rich in carbon and hydrogen, tend to be relatively non-polar. So these are some more detailed drawings of some of those lipids. So there's cholesterol shown up in the top. It contains a lot of carbon-carbon double bonds, and the carbons are organized into these ring structures. Those ring structures tend to be flat in 2 dimensions, and they are very rigid and not very flexible. And that's what lets cholesterol do the job that it does in membranes, which is make them a little bit stiffer so they hold their structure well. Too much cholesterol is bad, then membranes are too stiff and they tend to break and compromise cells. So you don't want to have too much cholesterol, but you don't want to have too little cholesterol either. You have to have just the right amount so that the membrane functions optimally at the temperature of the organism. Below cholesterol is just a designated free fatty acid. It's unnamed. And if you're unfamiliar with like the chemistry or like the conventions of chemical drawings, where you see these like little ridgy structures, those lines depict covalent bonds. Single lines are single covalent bonds, double lines are double covalent bonds, and everywhere 2 lines connect, there's a carbon. And also, there will be 1 or more hydrogens. So for each carbon, it has to have 4 covalent bonds. So I can see that, you know, some of them are connected to a carbon on either side, and then that would be 2 covalent bonds, and then they're also going to be an unwritten like hydrogen atoms written there. So this is a way of drawing these molecules really simply and in a clean manner that's not too cluttered for us to look at. But this structure going from left to right, that's the fatty acid tail. Carbon, 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 and lots of hydrogens. These are very non-polar. Below the free fatty acid, we have a triglyceride. And that has a similar structure, but it has an extra hydrocarbon tail on it. And then below that, we see another way of drawing a phospholipid. It's just oriented left to right. The fatty acid tails, the 2 of them are to the left, and the phosphate head is over to the right, and it's attached to an additional functional group, and we won't get into that too much, but that would be organism specific. Okay, so a little bit more on the waxes. So paraffin wax, which would be like you'd commonly see that in candles or be used for burning. And so would cycloparaffin and this other like isomer of paraffin. This is what wax chemistry looks like. So next time you see a candle, you should think about this. They're very hydrophobic because these molecules are full of carbon-carbon and carbon-hydrogen bonds, which are very non-polar covalent bonds. And they therefore do not inflict any areas of partial charge on the molecule. So there's nothing for water to hydrogen bond with. Many lipids are made of building blocks called fatty acids, and I already covered the chemistry of this, but I'm just going to point this out again. This is the other way of drawing. Like, see how cluttered this figure is? Because every bond is drawn in, and every hydrogen has been placed into the figure, right? And so that's one way of drawing these fatty acids. And another way of drawing them would be shown here with this like sort of wave-like convention. I should also add while we're looking at this that in general with lipids, if you have a lot of unsaturated fatty acids, those fatty acids will form liquids at room temperature. So think about olive oil or any other type of vegetable oil. It's a liquid at room temperature. And the reason that it stays liquid is these double bonds will impose a kink or a bend in the molecule. And so they tend to kind of keep some space between molecules and not pack together as tightly. And at room temperature, that keeps these substances liquid. In contrast, if you think about butter, butter is full of saturated fatty acids, as are animal fats, and they are solids at room temperature because this molecule, these saturated fatty acids stay very like straight, very linear, and they tend to then pack together a lot more tightly at room temperature, and therefore they form solids. So I'm going to introduce this one term and then close this section close this particular video lecture. I'm going to introduce this term called amphipathic. So think about the word amphibian, right? An amphibian is a creature that can live on land and in water. It goes back and forth. An amphipathic molecule, like a phospholipid, is a molecule where part of the molecule hydrogen bonds with water and part of the molecule does not hydrogen bond with water. In other words, part of the molecule is hydrophilic, the other part is hydrophobic. And phospholipids meet that criteria. The phosphate heads hydrogen bond with water and are therefore hydrophilic. The fatty acid tails do not hydrogen bond with water and are therefore hydrophobic. So there are like two natures, right, or tendencies, one water loving and one water phobic within this one molecule.